session, we are going to see the W chromosome for sexing of insects with special reference to silkworm. In insects, the sex is determined completely by the genetic mechanism, which at least in somatic tissues are the cell autonomers. The sex of the silkworm Bombyx mori is strongly controlled by the presence of W chromosome. Genetic studies using translocations and deletions of W suggested that a presumptive feminizing gene FEM is located in the limited region of the W chromosome. And recent genomic studies revealed that a small number of potential candidates for the FEM gene in this region. FEM RNA determines the sex of the female of the silkworm. FEM gene is specific to female silkworms, suggesting that it has a role in sex determination. Male-only rearing techniques for Bombyx mori are desirable because males show higher resistance to disease, lower food consumption, and better silk quality. To this end, several Bombyx mori strains for male-only rearing have been developed by classical genetics. Male silkworms weave cocoons with more silk for a higher quality than females do. And sericulture industry has a long sought and easy way to breed only males. Researchers have identified the process that determines sex in the silkworm Bombyx mori. The sex factor is found to be a small RNA molecule, the first time that anything other than a protein has been implicated in a sex determination process. In nearly all Lepidoptera, the order that includes moths and butterflies, sex is determined by the WZ chromosome system. In contrast to the XY system used in mammals, female silkworms carry W and Z sex chromosomes, whereas males boast a pair of Z chromosomes. The ZW sex determination system is reversed compared to the XY system. Females have two different kinds of chromosomes ZW and males have two of the same kind of chromosome, that is ZZ. Let's see the role of a FEM genes in sexing. The FEM RNA breaks down a corresponding molecule made by a gene known as masculinizer, which is found on the Z chromosome. When the researchers silence masculinizer, embryos execute a genetic program that makes female tissues. Male mutants of a moth die as embryos when even a tiny portion of the W chromosome is added to their gene. In addition, a Bombyx homologue of Drosophila sex determining gene double sex has been identified on an autosome and analyzed. Whereas the Drosophila double sex gene is regulated by activation of splicing in females. The Bombyx double sex gene BMDSX encodes female and male specific mRNAs regulated via male specific repression of splicing. The vitellogenin gene VG is a target of the BMDSX protein, which directly binds to the VG promoter. Furthermore, as ectopic expression of the male type BMDSX induces male-like transformation of the sexual organ, BMDSX may control sex-specific morphological characteristics in Bombyx. This suggests that although upstream events in Drosophila and Bombyx sex determination differ, similarities between the two species do exist in downstream genetic control of sex determination. Primary sex determination depends on a Z counting mechanism in Z bar ZZ species, but on a female determining gene, FEM in the W chromosome of the silkworm, the molecular mechanism is unknown in both sexes. The silkworm shares the last step DS6 of the hierarchical sex determination pathway in Drosophila and other insects investigated, but probably not the intermediate steps between the primary signal and DS6. The W chromosome is heterochromatic in most species. It contains few genes and is flooded with interspread repetitive elements. In interphase nuclei of females, it is readily discernible as heterochromatic body which grows with increasing degree of polyploidy in somatic cells. It is used as a marker for the genetic sex in studies of intersexes and Wolbachia infections. The sex chromosome system is being exploited in economically. Sex chromosome stand out from the rest of the chromosomes complement by being structurally different, having a different behavior in meiosis, sometimes being visible as heterochromatic bodies in the interphase nuclei, and by having an associated phenotype female or male. Since these basic observations are technically not demanding, sex chromosomes have been in the focus of studies from the early days of genetics and cytogenetics, and with ever refined research tools up to the present time. In Lepidoptera, in contrast to most other animal groups, females are the heterogametic cells. They share this property with Trichoptera, cadid flies, their close relatives, and with snakes and birds. Sex chromosome systems with female heterogamy are referred to as WZ bar ZZ and Z bar ZZ, alternatively designated WZ and Z0 depending on the presence or absence of the W chromosome. Vast amounts of sequence data have been acquired, including whole genome shotgun, that's called the WGS sequences of Bombyx mori, and express sequence tags, the EST tags collections from various species, developmental stages and tissues. A public database devoted to Lepidoptera allows researchers in the sequence collections and easy access to the data at www.butterflybase.org. The composition of the sex chromosome. The silkworm Bombyx mori genome sequenced independently by Chinese and Japanese groups and is publicly available from 2004. In both groups, male genomes were sequenced to avoid the complications expected from the W chromosome. Hence, the two assemblies include the contigs of the Z chromosome but not of the W chromosome. The Z chromosomal sequence contigs in Bombyx mori displayed no obvious general difference in composition between the autosomes and the Z chromosomes. Conventional genetic maps of the Z have been established not only in the silkworm but also in the European corn borer moth. Triphosphatase isomerase TPI endapterase were located in the Z chromosome of Bombyx mori. The period gene maps to the Z chromosome in Bombyx mori as well as in the Anthrea perni. Thus, there is an evidence for conserved synteny in at least a part of Z chromosome in Lepidoptera. Z chromosomal genes appear to contribute over proportionately to the speciation process. Pheromone traits, traits for male behavioral response to pheromone, and volatinism traits play an obvious role in reproductive barriers and are often Z-links, more often than expected under the assumption of the random distribution given the usually high number of the linkage groups in the Lepidoptera. Very few genes have been found linked to the W chromosome. The female determining factor FAM in the W Bombyx mori is the most prominent one, as said by Tazima. A Zion ectrite ESD is associated with the W chromosome, but its location has not been confirmed yet. Results obtained from the sex chromosome mutants of Epistia suggest that the presence of male killing factor in the W chromosome. In Papillogalatus, the dark morph trait is transmitted maternally and therefore considered W linked, but W linkages is disrupted in some crosses, presumably due to a Z-linked suppressor. However, the lack of association between the maternally transmitted mitochondrial DNA haplotypes and the W-limited phenotype make W linkages questionable. In Anthrea perni, several copies of the otherwise Z-linked period gene have been detected. One of them per W produces a truncated protein, while another one is the source of an antisense RNA, as said by Gotter in 1999. There have been several approaches to analyze the DNA composition of the W chromosome. Very general information stems from CGH, in which differently labeled genomic DNAs from the females and the males are competing for or are a female promoting maternal effect for by a gene on any chromosome including the W. This in fact is then a Z counting mechanism. A single Z chromosome causes female development and two Z chromosomes male development. Although no precise knowledge of the genes involved in generating the primary sex determining signal, both the dominating W and the recessive Z system do occur in Lepidoptera. Thus, it cannot be decided a priori whether sex determination in a WZ or ZZ species depends on a dominant W or a Z counting mechanism. The action of a Z counting mechanism was nicely shown in the cycad moth when the tetraploid females of a parthenogenetic race were crossed with a diploid male of a bisexual race. Triploid intersexes with the two Z chromosomes WZZ AAA resulted. All Z bar ZZ species necessarily have a Z counting mechanism. But again, it cannot be decided whether the male promoting activity of the Z is counteracted by a female promoting maternal effector, a female promoting activity encoded by zygotic autosomal gene Bombyx mori. On the other hand, represents the dominant W system. The W chromosome carries a female determining factor Fem. This has been inferred from polyploid and aneuploids. Whenever a W chromosome is present, a female develops from the embryo. In its absence, a male individual develops. This is in accordance with Tazima's findings. A couple of deletions and translocations of W chromosomal segments in Bombyx mori are known that they do not include Fem. The female determining function hence maps to the remaining segments of the W. But the precise location of Fem is not yet known, nor has Fem been isolated to reveal its molecular structure. Some information could be gathered from the back FISH in Bombyx mori. All autosomal backs that give specific signal on the respective autosomes and Z hybridized also to scattered sites on the W, according to the research findings of Yoshida. DNA sequences from the W, isolated as W-specific RAPDs, the random amplified polymorphic DNA in Bombyx mori, turned out to be truncated retrosposons as reviewed by the Abe et al. A direct approach, that is, cloning of microdissected W sequences, have been performed in the coding moth Cydia pomonella. Besides, a few non-coding unique W-specific sequences, non-coding and retrosposon sequences turned up, which were also present on the autosomes and the W chromosome, according to the studies of Fukova in 2007. Let's see the sex determination based on the primary signal it receives. In female heterogametic systems, the distribution of sex chromosomes to the egg decides upon the sex chromosome constitution of the zygote and subsequently sexual development of the embryo. ZZ embryos develop into males, while WZ embryos or Z0 embryos in Z bar ZZ systems into females. Since only one of the 4 nuclei that result from female meiosis is becoming the female pronucleus, the egg nucleus. The orientation of the WZ bivalent or the position of the Z univalent respectively in the metaphasal spindle determines the sex chromosome constitution of the egg. In a classical study based on Scherler research studies showed that the Z univalent behaves as a laggard in anaphase-1 spindles of the cycad moth, a species with Z bar ZZ sex chromosome mechanism. The position in the spindle near to the inner or to the outer pole is influenced by the ambient temperature. At high temperatures 30 to 37 degrees centigrade, the Z is more often found near the inner pole, thus more Z eggs are produced. At lower temperatures 3 to 8.5 degrees centigrade, it is more often seen moving to the outer pole and therefore more null 0 to Z eggs are generated. Consequently, the sex ratio among the offsprings is temperature dependent. Although the female heterogametic system has been conserved in all species so far investigated, the role of the sex chromosomes in the sex determination has not WZ systems are not different from the XY sex chromosome systems in this respect. Theoretically, the primary sex determining signal in a WZ by ZZ system may be generated by female determining genes on the W chromosome, while male development is the default. Thus, the presence of W causes the female development, absence of the W, male development, or male promoting genes on the Z acting against female promoting genes located on the autosomes. Let's see the ploidy level in male and females. Reading the table. At ploidy level 2N, females have WZAA while male has ZZAA and ZAA. At ploidy level 3N, females possess WW, ZZ, AAA, WW, Z, AAA, WW, ZZ, AAA, while males possess ZZZ and AAA, ZZ and AAA. At ploidy level 4N, females possesses WZZZ, AAA, further WWZZ, AA and AA. In males, it is ZZZZ, AA and AA. So, these are the different ploidy levels with the doses of W and Z chromosomes. Sexual development of polyploids and aneuploids in Bombyx mori based on the research studies developed by Kawamura, Shihara and Tajima, or a female promoting maternal effect which may be coded for a gene on any chromosome including the W. This in effect is then a Z counting mechanism. A single Z chromosome causes female development, 2 Z chromosomes male development, although we have no precise knowledge of the genes involved in generating the primary sex determining signals. Both the W dominant and the recessive Z system do occur in Lepidoptera. Thus, it cannot be decided a priori whether sex determination in a WZ or ZZ species depending on a dominant W or a Z counting mechanism. The action of a Z counting mechanism was nicely shown in the cycad moth Selenobia when tetraploid females of a parthenogenic race were crossed with diploid males of a bisexual race. triploid intersexes with two Z chromosomes WZZAAA resulted all Z bar double Z species necessarily have Z counting mechanism, but again it cannot be decided whether the male promoting activity of the Z is counteracted by a female promoting maternal effect or a female promoting activity encoded by zygotic autosomal genes in Bombyx mori. On the other hand, represents the dominating W system. The W chromosome carries a female determining factor FEM. This has been inferred from polyploids and aneuploids. Whenever a W chromosome is present, a female develops from the embryo. In its absence, a male individual develops. It is according to the research of Tazimer. A couple of deletions and translocations of the W chromosome segments in Bombyx mori are known that do not include FEM. The female determining function hence maps to the remaining segments of the W. But the precise location of FEM is not yet known, nor has FEM been isolated to reveal its molecular structure. Sex determination, the sex determining pathway. If you see in Drosophila, the primary sex determining signal, briefly called the X:A ratio, the ratio of X chromosomes to autosome sets, initiates a cascade of gene actions which ultimately regulate the development of sex-specific somatic characters. Sex-specific behavior and dosage compensation of X chromosomal genes based on the Mayer studies. The primary signal is in fact an X chromosome counting device based on the interaction of a couple of transaction factors. They are encoded by three classes of genes: X-linked numerator genes, an autosomal denominator gene, and maternal genes. The primary signal regulates the early expression of SXL, which denotes a splice regulator. SXL regulates its own expression in an auto feedback cycle, and in addition that of the TRA, the next downstream step in the cascade. TRA is again a splice factor which together with the TRA2 regulates the splicing of the DS6 transcript into the female or male specific form. Finally, the two isoforms DS6F and DS6M are transcription factors which can regulate the expression of the genes for the somatic sex-specific functions. This well-investigated sex-determining pathway of Drosophila had once been considered a paradigm of insect sex determination, but during the last 2 decades, it has become evident that only parts of the cascade are conserved in non-Drosophil insects. Among Lepidoptera, only Bombyx mori have been investigated with respect to sex-determining cascade genes. The ortholog of Drosophila's SXL gene is conserved in Bombyx mori, but the function is not. The XXL transcript is not sex-specifically spliced and hence probably not involved in binding sites of the chromosomes. From these studies, it is obvious that the intersped repetitive sequences abound in the W chromosome of most species. In some species, like Galleria, W-specific sequences predominate. In others, like Bombyx mori, the W chromosome has accumulated sequences equally present in males and females, presumably autosomal sequences too. From the above figure, sex-specific splicing of the Bombyx mori double sex, that is the BMDS6 transcript, exon 3 and 4 are skipped in the male supervariant. The coding sequence in the black boxes of the male-female specific variants ends in exon 4, that of the male specific variant in exon 5. sequence data from the phylogeny of the Lepidopteran sex chromosome systems. This is the data taken from the cladogram based on the Kristen and Karlsky.